So what we're going to talk about is kind of like the broad category of what we're talking about here at the end of our notes is genetic engineering. And so you could think of genetic engineering as a bunch of different things that can be done to alter the genes of an organism, to alter the DNA of an organism. As we've been talking about, our DNA basically tells our body how to work, what it looks like, how it functions. And so if we are able to alter the genes of an individual, we can maybe alter all of those things. So there's a few different ways this is done. So one type of technology that's been being done for 30 or 40 years is called gene splicing. This is when they basically extract some DNA, cut a piece out, and they could take that piece they cut from one organism and they could put it into another organism. And this has actually been pretty helpful, especially to people with diabetes. So people with diabetes need to give themselves what? Injections of, it is insulin, yeah. So it used to be, if you were diabetic, you needed insulin. The insulin used to come from pigs. They would harvest insulin from the pancreas of slaughtered pigs. And obviously, that was expensive. Some people had allergies, and it did not work as well as human insulin. But that's all they had for a while. And then what scientists, once this type of technology started to become well done, what they did is they took from human genes the instructions on how to build insulin, the gene that tells our body how to make insulin. They cut it out from the DNA and they put it into bacteria and then grew those bacteria. And what do you think those bacteria can do after that? And now they can make what? Yeah, they make human insulin. These bacteria, they grow them in huge vats, and the bacteria making, it's the exact same as insulin made in our bodies. And that's where all of the insulin that people take now, it comes from these genetic, okay? Oh, yeah, yeah. Yeah, it's a lot like that. Yeah. They grow these bacteria in huge vats, and then they can extract the insulin, and they have plenty of insulin. And so, and it works better than pig insulin. It's cheaper, and you can get a lot of it. So this was using this genetic engineering technology to produce that. And they've done this for other things like human growth hormone. They make it the same way. If a person has a lack of human growth hormone, they can put the human growth hormone gene into bacteria, grow the bacteria, and they produce the hormone. So that's one way that this gene splicing can be used. They basically transform bacteria so they can produce new products. It's been done in other ways too. They've taken a gene from bacteria that produces a chemical that repels insects, and they've put that gene into plants, corn plants, so that now those corn plants make this substance and insects are less likely to eat them. So they've taken genes from one organism and put them into another. So that's one way this is done, through gene splicing. And they can do this because we have these special types of enzymes called restriction enzymes. They cut DNA, and scientists can figure out exactly where they're going to cut the DNA. This allows them to kind of cut and paste. Yeah, this is a machine. I'll tell you what this is. So this is a DNA sequencing machine. So if they have a sample of DNA from somewhere, they can put it into a DNA sequencing machine, and it can tell you the order of the letters A, T, G, C, and so forth. And so that can be used actually to identify people. And that's what we'll talk about in a second. So here is an example. This is the process I just talked about. Here is a human cell. In the DNA, they find the gene for human growth hormone. They use a restriction enzyme to cut it out. They take the same restriction enzyme and they cut the DNA of a bacteria. They mix them together, and the human gene ends up in the bacteria DNA. They put it back into the bacteria and then grow those bacteria, and they can then produce, in this case, human growth hormone, or really any other human protein. The same process can be done, and it's relatively easy and been done many times. Well, it's not equipment. It's an enzyme. So it's basically a chemical. If you mix this certain chemical with the DNA, it cuts it at a certain base sequence. So like they don't have to see it. They don't have to like physically cut it. This enzyme does it for them. Sort of. Yeah. This enzyme, yeah, separates it, and these enzymes they look for a certain sequence. So, for example, one restriction enzyme might look for AAGCAA, and when it finds that sequence, it cuts it between the G and the C every time. Well, yes, these things. So, good question, Isaiah. Because let's take another example. There's a gene, BRCA1 and 2, that some people have. These are genes that make a person have a much higher risk of breast cancer. And some people have these genes, some people don't. You could inherit them from your parents. So, what some people would say, well, if we identify this gene, can we use this technology to alter and cut that gene out and replace it with a correct gene? I told you the other day I had a friend that had cystic fibrosis in high school. She died from that. It was a genetic disease that had, it's just one base in her DNA that when it was switched caused cystic fibrosis, which is when mucus builds up in a person's lungs, they get infections. If she, when she was a child, could have been, that gene could have been replaced with a correct gene, then it could have cured that disease that she died of. So there are instances, and scientists are working on ways that we can try to cure these genetic diseases that affect people. So when scientists do this and they take a gene from one organism and put it into another, what the result is what's called a transgenic organism. It's an organism that has DNA from another species put into it. Here are some examples of how it's been used. I already told you about insulin and human growth hormone. Those are a result of putting human genes into bacteria. Here's some others. Scientists have put human immune genes into mice, and then they can experiment on the mice instead of having to experiment on humans to test different medicines. They have put the gene for growth hormone into livestock, so they grow faster. Yes, yeah, so they produce more meat. They can take a gene for a certain medicine and put it into cows so that in their milk, they actually secrete the medicine in their milk and then it can be extracted. It's done in agriculture a lot where um, you could take a gene that produces an insecticide protein and put it into crops like corn or wheat or soybeans. And that's going to prevent insects from eating those crops. Um, putting Scientists put a gene that produces vitamin A into rice, and it produces a special type of rice that has high levels of vitamin A. That yes, it's the golden rice you see here. Yep, and it helps because a lot of people in developing countries where their main source of nutrition is rice, don't get enough vitamin A and it can lead to blindness. And so by growing instead this type of rice, which has extra vitamin A, it can help avoid that in these countries where you know have a good source. Not everywhere, no, just in certain places. Lots of times in Asia. But now there's the newest tool is this CRISPR that we were just talking about, or the video was about the guy in China that did the experiment on the two twins. Yeah, CRISPR is a way scientists have just developed in like the last 10 years or so of actually editing the genes in a living person. While there's so they can find a certain gene using this tool and they can change it in the cells of a living person. And so this is something scientists are really interested in because, like for example, I was talking about my friend with cystic fibrosis. If they could have gotten into the cells of her lungs and changed that gene, they could have cured that disease. Right. So if you can go in and edit the genes within a person's body, that has a lot of potential health um, advantages. It could be, and that's the thing. These things need to be studied a lot, right? Like, are there other risks? What does it mean? Because you're going to see in the later video today, they did this. I forget even what the disease was. A young man. What was it? I can't remember. Oh, you'll see it later. But they had a there when this is probably 15 or 20 years ago when they're first trying to develop these treatments. They gave this treatment to a young man who had a genetic disease, thinking it was experimental, and he actually died from this. And so for a long time, they stopped doing this research altogether because they felt it was too dangerous, and they started to do it more now. But that one situation sort of made people really think about, okay, well, what are we really doing here? Isaiah? Yeah, right. Yeah, and that part of what scientists have to do is figure a way to get that gene into the right cells and to try to change all of them. That is a good that's a good point. Another type of genetic technology that this used to be new, but now it's not. It's been happening for 30 years. Is cloning. Cloning is basically producing an exact genetic copy of an organism. The first cloned mammal was a sheep called Dolly was her name. It was 1997. Eventually, but here's how cloning works. Basically, they take a nucleus out of a cell from an organism, the whole nucleus. And remember, one single nucleus of a body cell has all of the information to make the whole organism. It's all in one nucleus. So they could take a nucleus out of one cell. Then they need to take an egg cell. They get rid of the egg cell's nucleus, so there's no genetic material. And they put the nucleus they took from a person or an animal, and they put it into the egg cell, and they zap it with a little electricity, and that starts it dividing. And then they take that embryo and they put it into the uterus of a female, and it grows. And eventually it's born, but that organism that's born is an exact genetic copy of the organism they took the nucleus out of. It's like an identical twin. Like, let's say if we wanted to clone Isaiah, we could take a cell from his skin, we put it under a microscope, we suck the nucleus out of it. Now we have a nucleus that has all the genetic information that makes Isaiah Isaiah. Then what we need is an egg cell that you can get from people donate egg cells all the time. So you can get an egg cell from a woman, you get rid of that nucleus because we don't want the woman's DNA in there. We want only Isaiah's DNA. We put Isaiah's nucleus in this egg cell, zap it with a little electricity. It starts to divide. It's a little embryo that's an identical genetic copy of Isaiah. They find a woman that will carry that. They implant the embryo into her uterus, and she gets pregnant. And she's pregnant for 30 weeks, right? And eventually she gives birth. But the child she would give birth to is Isaiah's identical twin. It has all of Isaiah's same genes, all of your same DNA. What? Only yours. And it's gonna when you were 1 year old, this baby is gonna look just like you when you were one. When this baby grows up to be 17, it's gonna look like if you had an identical twin, it's the same thing. Maybe, but here's the thing. Sometimes people think like cloning it like it's not Isaiah, right? Isaiah's still himself. It's just a copy of him. It's another person that looks like you. It's like you had identical twins and one was just born 17 years after the other. Maybe. And that would be interesting, right? Wouldn't it be interesting to see, okay, well, when this clone Isaiah 17, what's it like? Does it like the same foods that Isaiah likes? Does it like the same music? Does it have the same interests? Because if it does, that means, well, maybe that stuff is part of our DNA. Or maybe it's not. Maybe it likes completely different foods and music. And that tells us, well, probably those things are not controlled by our DNA, they're controlled by other factors. So that's called cloning. Another neat thing is a process that's done sometimes called electrophoresis. Electrophoresis is when you take some DNA samples and you chop them up with a restriction enzyme and you put them into this gel. This is what the equipment looks like. And you put them into this gel and you add electricity. And what happens is um, the DNA particles are attracted, okay, to one of the charges, the negative charge, and they start to move through this gel. You stop it after a certain amount of time and you stain it, and it forms a pattern. And this pattern that it forms can be used to identify people. Here is another picture of how it works. You put the DNA samples in these little wells. This stuff is like jello, basically. And small pieces of DNA can go through the gel really fast. Big pieces are slow because they have to work their way through all the holes. And so it starts to form a pattern of bands. So let's look at an example. Let's say this child was born from this mother, but she wasn't sure who the father of the child was. What do you mean? So what? So yeah. So you've seen those like shows where they're like going to reveal who the father is. The way they find out who the father is through this. Right. And so the child had to get each of these bands from either its mother or its father. And so if we look at this child, okay, well, this one could have come from the mother. This one came from the mother. This one came from the mother. These others couldn't have come from the mother, right? Because she doesn't have those bands. They had to come from the father. So if you look at potential fathers, okay, well, it couldn't be this bad one because he doesn't have one here. He doesn't have one here. He does have one here, but that couldn't be. He has to have all of the missing ones. How about this father? He's missing this one, right? He has others, but that's just maybe random. And if we look at this father, he has that one, that one, and that one. This must be the father. What else could this be used for? It can be used for crimes. The same process is used to try to identify people in crime scenes. If there's, for example, a blood, let's say there's blood at the scene of a crime. So you do the same thing. But then you have to have suspects. And if you say if the suspects, you take if you have a DNA sample from the suspects and you compare it to the DNA you found at the scene of the crime, that can indicate that person left that blood or a piece of hair or something else at the scene of the crime. So it's used. You can use this to match um, people to an unknown sample, and this can be made. These electrophoresis can be made with a very small amount of DNA. Sometimes, like if they're trying to collect DNA from a suspect in a crime, they might follow them around, and when they blow their nose, grab their tissue out of the garbage, or get their cup they drank out of at Starbucks and threw away, and they get from the saliva in that cup, they can get their DNA, and then they can use that to try to see if they also left their DNA at this crime in some way. Yeah. Um, I'm not gonna watch that. That's it. Okay. That's